Hello, and welcome to the 9th talk in the Mathematical and Computational Ophthalmology seminar series. My name's Paul Roberts, and I'm a lecturer in the Department of Optometry and Visual Sciences at City St George's, University of London, where I lead the Mathematical and Computational Ophthalmology group. I'm the creator and host of this seminar series, together with David Crabb, who heads the Crabb Lab at City St George's, University of London, and Phil Luthert, an Emeritus Professor of Pathology at the UCL Institute of Ophthalmology. This seminar series aims to showcase research in mathematical and computational ophthalmology to the broader ophthalmological, vision science, medical, mathematical, and computational communities, raising awareness of the exciting research being conducted in the field and to encourage future work in this area. Do subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. These are also listed on our worldwide page, the link to which can be found in the About section. Before introducing our speaker, Arto Erti, I'd like to highlight that the Investigative Ophthalmology and Visual Sciences special issue on mathematical and computational ophthalmology, guest edited by David Crabb, Phil Luthert and me, is almost complete. 35 papers have been published thus far, including an invited review from Arto, with a few more to follow. A link to the special issue can be found in the About section. I coined the term mathematical ophthalmology to denote the field which uses mathematical tools to preserve vision by elucidating the mechanisms underpinning ophthalmic health and disease, while computational ophthalmology is an existing term used to denote the field which uses computational algorithms, in particular AI, to preserve vision by identifying and predicting ocular features and risks. Thus, the focus of mathematical ophthalmology is upon elucidating mechanisms, while that of computational ophthalmology is upon prediction and identification. In our last seminar, Christine Curcio gave a fantastic talk on why age-related macular degeneration is a mathematically tractable disease, the recording for which can be found on our YouTube playlist, together with recordings of all previous talks. This week, we're joined by our speaker, Arto Erti. If you'd like to ask Arto a question, then please do send these in via the YouTube chat both during and after the talk. The talk will last for about 45 to 50 minutes, followed by 10 to 15 minutes for Q&A, in which I will relay your questions to Arto. So it gives me great pleasure now to introduce Arto Erti. So Arto is Professor of Biopharmaceutics at the University of Eastern Finland. His research is focused on issues of ocular drug delivery and pharmacokinetics, spanning pro-drug discovery, formulation technology, including nanomedicines and controlled release, biological testing in cultured cells, tissue and animal models, advanced analytics, as well as in silico modeling. His research has been published in more than 400 peer-reviewed articles that have been cited over 26,000 times, with an H-index of 83. Further, Arto was ranked in the top 0.05% among global researchers in the field of drug discovery. He is co-inventor of 25 families of patents and patent applications. His research is funded by the Research Council of Finland, Business Finland, the US FDA, the European Union, the Sigrid Juselius Foundation, and several SME and big pharma companies. He's also received various awards for his research activities in Finland, the European Union, the United States, and Japan. So I'll hand over now to Arto to present his talk entitled Computational Modeling of Ocular Pharmacokinetics. Arto.
Okay, thank you, Paul, for this invitation and kind introduction. And what I'm trying to do today is to tell about the mathematical models of ocular pharmacokinetics. And you have my contact address here. And if you have, after the lecture, any questions or would like to contact, feel free to do so. So the contents of this lecture will be first about some basics of ocular pharmacokinetics, model types, and then I will discuss the most important routes of drug administration to the eye, which are topical delivery, intravitreal, and systemic, and some aspects of the modeling in that context. Then finally, something about melanin binding, which is a peculiar feature in the ocular pharmacokinetics, and interspecies translation, which is quite important in drug development. So when the eyes are treated with medications, most often the drugs are given in a topical way, local administration, meaning that these drugs are given either as a topical eye drops on the surface of the eye, where they may absorb into the eye through the cornea and conjunctiva, but this is a way to treat the anterior segment of the eye, like a cornea, iris, ciliary body, trabecular meshwork, but not the back of the eye. In order to treat the back of the eye, intravitreal injections are given to reach the retina and choroid, for example, in the treatment of AMD. Then there are other routes of administration, like systemic route, but that is not usually as feasible because higher drug doses need to be given. So the anterior segment treatment is a home treatment with eye drops. It's non-invasive. These are attractive features. Then the limitations include the poor absorption of biologics, for example, so that it's limited to the use of small molecules. Widely used for reduction of intraocular pressure in glaucoma. The posterior segment treatment with intravitreal injections mostly. There are some other injections also, like suprachoroidal and subretinal, that I will introduce as well. Is invasive, obviously. It's used clinically routinely in the case of intravitreal injections, but the other injections are more specialized. It is feasible for small molecules and also for biologics and in emerging gene therapies. Indications include, for example, diabetic macular edema, AMD, uveitis. So these other specialized injections include, for example, subretinal injection between the neural retina and retinal pigment epithelium that is used, for example, in the gene therapy of the eye, Luxturna being the commercial product, and suprachoroidal route between the sclera and choroid, which is more like an emerging way of delivering drugs to the eye. Subconjunctival injection has been investigated for retinal delivery, but the retinal delivery is relatively poor, but it's used in some cases to treat the anterior segment of the eye. So if we take a look at the basic features of ocular pharmacokinetics before getting into any models, there are several anatomical barriers that are limiting drug penetration. On the surface of the eye, there's the cornea, especially corneal epithelium, which is this thick black line here. In this figure, the continuous lines indicate penetration barriers for drugs. Then the dotted lines indicate more open interfaces for drug penetration. So in the cornea, there is this epithelium and stroma, epithelium being the barrier. In the conjunctiva, there is the barrier of conjunctival epithelium. Then inside the eye, there is blood ocular barriers consisting of ciliary body blood vessels, retinal capillaries, and retinal pigment epithelium, and then the epithelium in the ciliary body. These are limiting drug penetration between the eye and blood circulation. The highest blood circulation in the eye is in the choroid, in the choroidal vessels. This is clearly the highest blood flow in the eye, but these choroidal vessels are quite fenestrated and leaky, so that the drugs that are in the systemic circulation can escape these vessels, but then they are limited. They have limited access to the neural retina and other parts of the eye because of the barrier of retinal pigment epithelium. So then in the case of the ocular, the topical administration, there's also the flow of the drug in the tear fluid to the nasolacrimal duct. in the aqueous humor, there's aqueous humor outflow that is contributing. And then, of course, there are different blood flow factors that are influencing pharmacokinetics. In addition to these anatomical and flow factors, there are different biochemical features, receptors, drug metabolizing enzymes, and transporters. In this lecture, I will not discuss much about these receptors, metabolizing enzymes, and transporters because their role in ocular pharmacokinetics is relatively unclear still. It's known that the drug metabolizing enzymes usually contribute quite little to ocular drug elimination. Rather, different other factors that I will tell are much more pronounced. And the role of transporters is still emerging and therefore not quite clear yet. So what kind of challenges there are in ocular drug administration or drug delivery? First of all, in the case of topical delivery, as I said, only the anterior part can be effectively treated. Also, the duration of action of these small molecules is relatively short in the eye, so that these eye drops are given at least once daily, often multiple times daily, and that will reduce the patient compliance, which is low, for example, in the glaucoma, even less than 50%, which is very bad because then the disease is progressing and it may harm the patient's vision. Systemic delivery has also some issues. First of all, the difficulty to getting across the blood-ocular barriers, so that relatively high doses would be needed in systemic administration, and of course that is exposing the patient to systemic adverse effects. Intravitreal delivery is effective in terms of delivery. The problem is that it's invasive and it's a burden to the healthcare, and more extended injection intervals would be very much wanted to minimize these visits and injections. Also, there is some uncertainty related to the retinal delivery of some forms of medications. So this ocular pharmacokinetics is highly related to drug delivery and drug delivery systems that are designed in order to optimize this pharmacokinetics of a drug. For example, regulating the concentration levels, extending the duration of action, and enabling less invasive modes of delivery. I have classified them into two categories here: controlled release systems that are macroscopic in nature, so that they are in the scale of millimeters in size, and they are releasing the drug at the site of injection or site of application for prolonged times, usually at least weeks, often months, in some cases more than 1 year. And they should have a long retention at the site of injection. For example, here is an Ozurdex, which is a biodegradable dexamethasone implant injected into the vitreous, where it releases the drug over some months. And this is anti-inflammatory medication. Another example is Durysta, which is intracameral small implant releasing ocular pressure drug brimonidine over long times, thereby offering an alternative to the more frequent eye drop instillation. So these are used to release small molecules. In principle, they could be used for biologics also, but in the current clinical practice, such products are not yet available. So these systems are passive in a sense that they are releasing the drug, and then the fate of the drug is defined by the pharmacokinetics of a free drug. Targeted drug delivery systems have a different principle. Intention here is to change the drug distribution between different tissues and cell types by using nanosystems that are much smaller. So they are now in the scale of nanometers, and they are attractive, for example, for DNA and RNA-based therapies. One example is liposomes, which include these lipid bilayers, and you can encapsulate drugs inside. Another example is lipid nanoparticles that have become famous for messenger RNA delivery in the case of COVID-19 vaccines, but these kind of technologies are attractive also for ocular treatments. Here, RNA is bound by ionizable lipids, and then there is some tailor-made lipid mixture to optimize the delivery properties. So why then modeling? So traditionally, pharmacokinetics in the eye is studied in animals, primarily in rabbits. And that is because obvious reasons you cannot sample human eyes for measuring drug concentrations. That would be too invasive and ethically not acceptable. So we have only very limited data from humans in terms of ocular pharmacokinetics. So in these rabbit studies, individual parameters are often determined. There's a lot of literature values for permeability coefficients of different drugs in the cornea, for example, or you can have in vivo application of eye drops or injections, and then people have measured concentrations in different tissues. And then that is fine, but then the problem is that this you cannot from such simple studies predict what would be effect of dosing, formulation, drug release, or disease state, changes in certain permeability of a membrane, for example, what could be, how it could be affecting the overall situation. And also the interspecies scaling has been lacking, so that how to scale from mouse to rabbit or scale from animals to humans is not available. And that's problematic because in the design of clinical studies, you would need to decide what kind of dosing would be most relevant in humans. So models have some advantages. So first of all, you can make an integrated system that would describe pharmacokinetics, taking into account several parameters simultaneously. It could predict different situations so that if you are changing one parameter, would the system be very sensitive to that or non-sensitive? That would be quite important in drug development. And then also, if you are changing chemistry of the drug or formulations, how that would impact the pharmacokinetics and perhaps also drug responses. And of course, then this the human translation and interspecies scaling is quite important. So we can divide these pharmacokinetic models to two basic categories, top-down and bottom-up models. And idea of the top-down model is that you would give the drug to the animals. You can measure the drug concentrations in different tissues, and then you can describe this using non-compartmental system or compartmental models, or perhaps population pharmacokinetics. You would get different values like clearance and volumes of distribution and so on. That would be then useful to build bottom-up models where you can simulate effect of different factors. For example, physiologically based models, finite element models, semi-mechanistic models are in this category. These could be utilizing also in vitro parameters that you would obtain with isolated membranes, for example, and you could include different physiological factors like flows or receptor affinities to predict drug responses. So these kind of predictive bottom-up models are quite useful. Oftentimes, like in the many examples that I will show, there are kind of hybrids, so that there are some features that are obtained from top-down approach and then something that can be obtained from in vitro studies, some parameters, to build a model that includes many, many features. The aim in our research is very much to predict, because we do preclinical research. We want to predict what's going on in humans, and also from in vitro to in vivo situation to get kind of a more kind of a solid background on drug development. So here are some features of the kinetic model types that are being used. Compartmental models are usually used in a top-down approach. You have concentration gradients, and using differential equations, you end up having different parameters solved, and then you have a curve fitting to find the optimal curve fit and the associated parameters. Population PK/PD has been very little used in ocular pharmacokinetics, and I'm not talking much about that, but the advantage here is that you can use sparse data, so that in a compartmental model, usually you have many time points in a pharmacokinetic study, and then you make a curve fit. In a population model, you may have a much larger number of subjects. It could be even hundreds, like from a hospital setting, for example, but you would have only one or two concentrations from each one. And then population model is pooling them all together and giving some variability and confidence limits for these parameters. Physiologically based pharmacokinetic models are integrating physiological features into the model. These two compartmental and population models have some kind of artificial or virtual compartments that they are dealing with. PBPK is trying to mimic the physiology of the body in different species, and this is useful especially for interspecies scaling, because the same model structure could be used for the one species and then converted to another species by changing the numbers in the parameters, for example, size of the vitreous and so on. And then CFD, or computational fluid dynamics, or finite element models, are more complex. They can include thousands of compartments in the eye, and then you define movement from one compartment to another. Difference compared to the other models also is that this is, first of all, very like a heavy computing. It has a lot of numbers, but it can provide also concentration distributions within tissues. So it is much more microscopic in a way as compared to these three other model types. So compartmental models has been the most traditional one. It includes a compartment, and then based on the kinetic in vivo data, you solve these differential equations for one or two compartment models, and then you can obtain clearance, volume of distribution, and so on, half-life. Usually it's used for one and two compartments. Of course, you can increase the number of compartments, but then you make a curve fitting. If you have 5 compartments, then the credibility of the individual parameters that are obtained becomes a bit questionable. So usually the number of compartments is kind of minimized for the purpose of the good fitting. And this has been used for the eye for a long time. For example, if you give intravitreal injection and then there is an elimination, you can define volume of distribution for the drug in the vitreal compartment and the clearance value from the eye. Population pharmacokinetic model has a kind of a similar structure as the compartmental model, like here, but it takes into account the statistics, the variability between different patients, for example, and then you can get the covariates, like how is the weight or some disease affecting the pharmacokinetics and so on. Limitation with this in the case of ophthalmology is that the pharmacokinetic data from humans is very sparse, but on the other hand, response data of drug treatments is available. So the PD part would be quite feasible for this population approach, but linking kinetics and responses becomes difficult because there is not so much of the PK pharmacokinetic data. Physiologically based pharmacokinetic models have been traditionally built like this, whole body PBPK, and there are some software packages by Certara and Gastro Plus that include this kind of a whole body kinetic models. There are a lot of data, different in vitro parameters, physiological factors like blood flows, partitioning of the drug from to different tissues, kidney functions, and so on. Everything can be included. And the nice thing with this kind of a model is that you can translate from rat to human, for example, and industry is using it for that kind of a purpose. In the case of the eye, there's much less available as compared to the whole body PBPK approach, but in principle it could be built for the eye, and there are people who are doing this as well. In this case, of course, the ocular volumes, flow factors, partitioning to different tissues, or their predictions will be included in such a model. And then finally, finite element models have a lot of these different compartments, for example, here in the vitreous. And then you can define the diffusion within the vitreous, diffusion in the retina, different flow factors, and so on, and trying to make up like a virtual eye where you could predict what happens when you inject something into the eye. Then something that is quite powerful and useful to be connected to these pharmacokinetic models is a quantitative structure-property relationship. So the idea here is that you would have a certain parameter, for example, conjunctival permeability. You can measure that in vitro with isolated conjunctivas, and then you could have a number of compounds. You study the permeability and then you try to correlate that with the chemical structure. Oftentimes a single parameter is not enough, so that, for example, here log D value, indicator for lipophilicity, is plotted with the conjunctival permeability, but what we can see is that the correlation is not that good, and simple lipophilicity is inadequate to describe it. But if we have a large number of different chemical descriptors, and then in a process of QSPR model building, you kind of find out which are the most critical physical parameters, then you can end up to this kind of a simple QSPR models. For example, here we have PSA, which is the polar surface area of the molecule. This can be obtained computationally, and hydrogen bond donor groups, which can be obtained also computational. So then with the computational means, you could get estimate for conjunctival permeability even without any testing. And such predictions are quite useful because you can use them as a part of your pharmacokinetic models to build the models, so that you don't need to get experimental proof of every value to build a model. So that can save a lot of time. Here is an example of the experimental conjunctival permeability values compared to the predicted ones with QSPR model in internal test set and external test set, and they show pretty good correlation. So this is something quite useful to be combined. And of course, it can be this is used also for predicting drug responses on receptor affinity and so on. Okay, so then next I will describe some administration routes and modeling approaches related to them, just selected modeling approaches, not comprehensively. First of all, topical delivery, usually eye drops, sometimes ointments used. And in that case, the drug will be mixed in the tear fluid. And then it has several factors that are moving the drug out from the tear fluid. There's normal tear flow, but even more important is the drainage of the extra fluid. It goes to the nasolacrimal duct and then to the nose. And then there is a systemic absorption across the conjunctiva of the eye. Also from the palpebral, the lid side, there's a rich blood flow, and that is removing quite effectively the drug from the tear fluid. Actually, much more goes there. Often, for example, in the case of timolol, 80% goes to systemic circulation and about 4% goes into the eye. Usually, the bioavailability of the small molecules to the eye is from 0.1 to 5%, as determined based on the concentrations in the aqueous humor. Partitioning into the corneal epithelium is the first step in the absorption, but this one is competing with these loss processes. And because these loss processes are quite effective, we will have such a small bioavailability. After partitioning to these epithelia, then there's a diffusion across the corneal stroma and endothelium to the aqueous humor. And from aqueous humor, the drug is eliminated by normal outflow of the aqueous humor, but also distributing to the iris ciliary body and removed from there by the blood flow. This factor can be higher than this normal outflow. So any drug can get out with the outflow, but if the drug is lipophilic and small, it can be very effectively also removed by this blood flow in the anterior uvea. This can be even tenfold compared to the outflow elimination. The other route inside the eye is through conjunctiva, drug partitions to the conjunctiva, then first to epithelium and then diffuses across conjunctiva and sclera to iris ciliary body, and there it may have its effect, and also it can be eliminated. There is also blood flow related elimination within the conjunctiva. So overall, actually this absorption from eye drops is quite a complicated process to prediction. For example, permeability studies are done in some kind of a steady state situation in diffusion cells with isolated membranes. But in the in vivo situation, there is initial partitioning to the epithelium and then after that, slow diffusion. And that's more complex for modeling. The outcome in the eye drop administration is that there are huge differences between concentrations in different tissues. Here we have a study where we investigated 3 different beta blockers: betaxolol, the most lipophilic one, timolol intermediate, and atenolol is the most hydrophilic compound. On the y-axis, we have AUC values, area under the curve of the concentration versus time in each tissue. So this is drug exposure to each tissue. And you have different tissues on the x-axis. So this represents actually quite huge data. So if we take a look at the y-axis, it's a logarithmic scale. So starting from the tear fluid, we go several orders of magnitude lower when we are going to vitreous humor. So very, very little of the drug ends up to these posterior tissues. and there is a quite steep concentration gradient over a short distance, describing how effective these ocular barriers are in limiting further distribution within the eye. Also, you can see that the more lipophilic drug, petaxolol, with these crosses, has usually higher exposure to these tissues, timolol being in the center, and then atenolol having the lowest levels. There are in the literature different models for topical ocular drug delivery. What I would like to introduce here is a relatively simple and practical approach for drug development, which is estimation of average steady-state concentration in aqueous humor. So this is a general equation used also for oral medications. So the required topical dose here is defined by what is your targeted average steady-state concentration in the eye. And that is, of course, dependent on potency of the compound, how much drug would you need to have the activity in the anterior chamber. Then the clearance from aqueous humor from 5 to 35 microliters per minute, based on the literature. Lipophilic compounds tend to have higher values, hydrophilic less so. Dosing interval is tau, and then you have bioavailability from 0.1% up to 5%. And then basically the clearance and bioavailability user should predict somehow based on the literature to get some kind of a rough estimate, and then you could calculate what dose you would need to reach certain steady state levels during multiple dosing regimen. And then depending on the target concentration that you wish and the dosing interval, you can estimate the dose in each eye drop. So this is very simple and effective way to make such decisions. So next I will move to intravitreal injections, which are very important in the clinical anti-VEGF treatment, and also in particularly in the neovascular AMD and in diabetic macular edema. These injections are given usually at 1 to 2 month intervals, and these are protein drugs like antibodies and soluble receptors. There would be a need for longer injection interval that would be beneficial for patients and the clinics as well. So after this injection into the vitreous is some kind of like a hydrogel type material. The drug is diffusing there in all directions, also towards the retina to exert its action. And in principle, the drug could escape from the eye by transretinal permeation, which is posterior route, or anteriorly going to the aqueous humor and then with the aqueous humor flow getting out of the eye. So the posterior elimination is possible if the drug is capable of penetrating these membranes, like ciliary epithelia and retinal pigment epithelium, capillary tight junction walls of the retinal capillaries, and then the vessel walls in the ciliary body. Protein drugs are not so capable of penetrating these barriers, so therefore they stay in the eye for a longer time. And that we can see here in this graph. So if we investigate half-life of different protein drugs in the eye, the half-lives tend to be in the range of 100 hours or more. So it's like many days, up to say to 10 days. These are in rabbits. In humans, the values are a little bit longer, but relatively similar. For small molecules, these blue and red dots, the half-lives are much lower. So they tend to be ranging from, let's say, from 1 hour to maybe 20 hours, most often a few hours. So that's very short. And that is because these small molecules are capable of crossing these posterior barriers. So therefore, they disappear from the eye more quickly. And the clearance from the vitreous agarose blood-retinal barrier is defined by this permeability in the membrane, in these membranes, permeability P multiplied by the surface area of these membranes. So if there are multiple membranes, there are PS terms, several of them summed up. So the elimination of the protein drugs is only anteriorly because this posterior elimination is so limited. So they will diffuse also to the retina, but the mass balance, the most of the drug mass, is diffusing to the posterior chamber and then to the anterior chamber, and with the aqueous humor outflow it goes out. So this is much less effective way of removing drug as compared to the posterior elimination. So therefore the half-lives are much longer. And if we compare these values to the blood flow, here is a choroidal blood flow in the eye, which is very high. And if you compare even these small molecule drugs, the clearance values, they are much below the blood flow, indicating that there is some barrier, membrane barrier that is limiting the elimination. So it's not blood flow limited. Also, if you look at the aqueous humor outflow value, this blue dotted line, and compare the clearance of large molecules to that value, these molecules are diffusing, eliminating from the eye clearly at a lower rate. And that is because the drug doesn't have a full access to the aqueous humor outflow immediately. It has to go through these small channels between the lens, impermeable lens, and iris to get into the aqueous humor for elimination. Volume of distribution in vivo determined with compartmental analysis tells us that this volume of distribution has very narrow range, so that it's very close to the anatomical volume of the vitreal cavity in the rabbits. And then finally here in this graph D, we have a clearance that would be defined by permeability of RPE multiplied by the surface area of the RPE. If we calculate from in vitro studies the permeabilities for FITC dextrans macromolecules from 4 kilodaltons to 80 kilodaltons, we have these small values. The experimental values are clearly higher, and that is because they are actually not, they would have so small clearance if they would be eliminated by posterior route, because the permeability is so low in RPE, for example, but they are eliminated anteriorly, so therefore they have this kind of a much higher clearance values, even though they are much lower than the values for small molecules. So how we can model these intravitreal injections? If we inject the drug into the vitreous, and then we can estimate the clearance from vitreous into the anterior chamber, taking into account the surface areas, the volumes, and the diffusivity. And then we assume that from the aqueous humor, the outflow determines the clearance. And then we can estimate that the posterior clearance is 0, or it is only 10% of the total clearance. So we did that kind of a simulations. First of all, if we simulate the intracameral elimination, so that the drug would be injected to the anterior chamber and removed by aqueous humor outflow, the curve would look like this. Very steep decline, very rapidly. But in reality, ranibizumab, for example, in aqueous humor looks like this. And that is because the ranibizumab injected to the vitreous is gradually coming to aqueous humor. What comes to aqueous humor is rapidly eliminated. So then the distribution from vitreous to aqueous humor determines the kinetic, and then therefore it's much more shallow. In this graph on the right side, we have 5 different biologics that have been injected into the vitreous, and then this solid line describes the simulated vitreal concentrations, and the blue dotted lines describe the kinetics in the aqueous humor, and the symbols are the experimental data for these 5 biologics. As you can see, the levels in aqueous humor, as expected, are lower than in the vitreous. Also, you can see that the experimental data and the simulations match each other quite nicely, telling us that this elimination indeed takes place anteriorly. Both vitreal and aqueous humor data are matching with the simulations. So then we did a similar kind of exercise with liposomes, which are having a size of 50 nanometers. So they are bigger than antibodies, for example. We gave intravitreal injections to the rabbits and used in vivo fluorophotometry to follow the kinetics of these labeled liposomes in the vitreous, which is these symbols, and these green symbols indicate the levels in aqueous humor. Again, much lower levels in aqueous humor, but again you can see that the slopes of these 2 lines are very similar, indicating that the aqueous humor kinetics is controlled by vitreal kinetics. Again, the simulated model described here is matching with the experimental data quite well. Again, here we assume that everything is eliminated by anterior route and no liposomes would go directly across blood-retina barriers to the blood circulation. And this model structure supports the experimental data and gives us information about the route. For example, this kind of issue would be quite difficult to study experimentally, where it goes, but simulation can give this kind of important answers. Then some words about the finite element models for injectables. First, intracameral injection of hyaluronic acid. In this kind of a finite element model, we can have other features, not only concentrations, but also, for example, ocular pressure and the temperature, localized temperature in the eye. So this shows in the finite element model the temperature in the surface of the eye, and then it's a little bit higher when you go deeper into the vitreous. If we take into account this temperature gradient, it will cause some fluid mixing. Without heat transfer, there is very poor mixing, and if you inject the drug to this anterior chamber, the drug is mixed with the aqueous humor flow, with aqueous humor very slowly, and then the elimination from anterior chamber becomes unrealistically slow. But if you include this heat transfer, then the mixing takes place, and then the elimination becomes very realistic. Note that in these kind of more simple models that I have presented, we assume that once the drug goes into the aqueous humor, it's mixed immediately. So it's like a well-mixed tank in that sense. Here is another example of finite element modeling, now intravitreal injection. An intravitreal injection was given, and then we are assuming partitioning to the retina using partition coefficient of 0.5. and we were able to match the data for vitreous and retina and aqueous humor, all of them in the same simulation. But this is quite complicated. It's I think it's a way to build a virtual eye in a very complex manner, but it requires a lot of details. And for some practical things, more simple models can be effective enough. Then one aspect is how we can use this kind of a modeling in a drug delivery system design. So we have here from our paper in 2015 some ideas about that. So we collected the data for all data at that time, something like more than 50 compounds that were injected to the rabbit eyes intravitreally, and then we curated the data to check the reliability, and then we could make a computational prediction of the clearance. So this was a QSPR model where we had some physical parameters that describe clearance from the vitreous. And then we used these parameters to pharmacokinetic simulations that you could then predict concentration profiles if the drug is formulated to some controlled release implant, for example. And then we could simulate the concentrations in the vitreous as a function of dose, release rate, retention of the system, and so on. For example, here is the concentration simulated for an implant that has a certain release rate and a dose, and then we have either high clearance or low clearance drug, and then you can see that the levels are quite different. So then these kind of levels could be compared to the target concentrations, and in that sense, it would help the design of these controlled release systems. Drug loading is another issue in these systems. For example, if you have a 3-month device so that it releases the drug at first order rate kinetics for 3 months, and it's given repeatedly to the patients. Then here is the minimum concentration during the multiple dosing, and then the dose per injection is here. So if you would have a drug that has a 10 micromolar target concentration in the eye, you would need to have something like 10 milligrams of the drug in the injection. And depending on the formulation type, this can be quite challenging and difficult. In some cases, basically impossible. And then to know this would be very beneficial in drug development, could save a lot of effort. But if your drug would be very potent, so that 10 nanomolar would be enough, then you would need very little drug in each injection, something like 10 in the range of 10 micrograms. And that's a completely different situation. So modeling can be quite helpful in describing and understanding the kinetics in the vitreous and its elimination from the eye. But then how about the retinal kinetics? So in the case of retinal kinetics, the field is not as advanced. We know that small molecules can diffuse in the vitreous. They can get across the inner limiting membrane to the neural retina, and depending on the compound, they can also get across the RPE to be eliminated via choroidal blood flow into the systemic circulation. In the case of biologics, again, they can diffuse in the vitreous. They are known to penetrate across inner limiting membrane, get into the neural retina, but the biologics are not getting effectively across the RPE or not getting through retinal capillary walls. So therefore, they are not eliminated in the retina, but rather they back diffuse later on to the vitreous and go out anteriorly, which is prolonging their retention in the eye and their activities. In the case of nanomedicines and also viral vectors, they have more restricted diffusion in the vitreous, even more restricted in an inner limiting membrane, depending on the formulation. Anyway, and then they don't get across the RPE. So they may stay long times also in the retina. But quantitative information, experimental information is quite sparse still, and we don't understand the kinetics within the retina. In order to understand the kinetics in different retinal layers, which could be quite important, for example, for nanomedicine's case, the verification would require excellent analytics with high spatial resolution to get data about these concentration gradients and localization and targeting and so on. But we are not there yet. One aspect related to this retinal kinetics is also the systemic delivery. So if the drug is coming from systemic circulation, it comes to the choroid, can easily escape from the choroid, but then it has difficulties to get across the RPE. In the case of small molecules, the unbound drug in the plasma can go across blood-retina barrier into the retinal tissues. So here we have, this is actually a human study. Patients have been receiving statins, and the concentrations in the blood circulation in plasma are these red symbols for different statins. There's simvastatin, atorvastatin, rosuvastatin, for example. This is a chronic multiple dosing. And then the concentrations in the vitreous are these blue bars. So it is the range, you can see it's much lower, at least 10, maybe 100-fold lower than the levels in plasma. So then if we are trying to model and calculate the vitreal concentrations, we use this kind of a model where we are assuming that only the unbound drug gets into the eye and the protein bound, albumin bound does not. And then these gray squares describe the levels of the results of these calculations. As you can see, they are pretty much in the same range as these measured concentrations in the vitreous. And indeed, the protein binding is very important parameter here in defining how well the drug can go from blood circulation to the eye. Another important feature in the ocular field is melanin binding. Melanin is a pigment that is found in iris, ciliary body, retinal pigment epithelium, and choroid. And because there's a lot of pigment, it may affect pharmacokinetics, because it's known that some drugs are bound to melanin, which is inside the melanosomal membranes. So melanin is like a very tiny particle, and drugs may bind it, and there will be an equilibrium of bound drug and free drug, and it may form some kind of a depot that is releasing the drug gradually. And this can be quite relevant for different delivery routes because this pigment is both in the anterior part and in the posterior part. And there's a lot of this pigment, and it can have really an impact. So this impact may be prolonged retention in the pigmented tissues and a prolonged drug response. We have been investigating for about 10 years more systematically the pigment binding to understand it. We did in vitro binding studies, and what we found out was that this kind of a CIPS isotherm mathematically better describes the binding as compared to Langmuir isotherm or Scatchard plots, for example, because it seems that there are multiple binding sites, so that for each drug they may bind in different ways to melanin surface. So there is not defined binding pockets like drug receptors have. But melanin, even though melanin has the binding has relatively low affinity, but the capacity of melanin is very high. So therefore it can have a big impact and form this kind of a depot and drug accumulation. Here is an example from an in vitro study. levofloxacin is a high binder. It was given intravenously to the rats. And as you can see in the pigmented rats, the retention in the eye is very high, while in the albino rat, the drug disappears very quickly. And then if we have a celecoxib, which is a low binder, then their kinetics in the eye is rapid in both cases. In plasma, no difference between two rat strains. So it can really have a big influence. So this long retention in vivo was shown in the previous slide. But then if we want to understand why there is such a long retention, it becomes a little bit more complicated. For example, if we make a study where we take these melanin granules, incubate them with the drug, and then remove the medium with the drug and replace it with the blank medium and study the release, we observe that the drug is released in maybe 30 minutes or 1 hour very quickly, like this, the red line here. If we have a melanosomes isolated so that they have this lipid membrane around it, then there is some extension of the release. But if we have a pigment-loaded cells and like RPE cells and then study release of the drug from those cells to the medium, then it lasts something like 10 days. So there's a remarkable extension. So why is that? It means that this melanin binding itself is not keeping the drug. It's more like an interplay of a melanin-bound drug, and there will be an equilibrium inside the melanosomes, and a very small fraction of the free drug is capable of permeating across melanosomal membranes to the cell cytosol, and then from across plasma membrane to the outside of the cell. So there is a complex interplay of these different factors. And that can be modeled actually. And we have done such a model using parameters for chloroquine as a high binder compound and timolol moderate binder compound. And these are the binding features from CYPS isotherm for these two drugs. And then we can follow, if we assume we give intravitreal injection, we can follow the drug in the RPE choroid, total drug, and also the free drug in the RPE choroid. And here are the concentrations, and this is for chloroquine. So you can see that it retains very long time. So the scale here is 60 days, but there's still a lot of drug remaining. And the elimination is related to the membrane permeability. So if we assume in the simulation that the melanosomal and plasma membrane have this kind of a permeability, 10 to minus 5 centimeters per second, the drug is eliminating faster than in the case of 10-fold slower permeability or 100-fold slower permeability. The dotted lines describe the unbound drug in these three cases, and as you can see, they are much lower than the bound drug. So only tiny fraction is actually unbound state. That enables the long retention, but on the other hand, if you want to have a prolonged drug response, you need to maintain the drug at the effective concentration. So this means that if the drug is very potent, for example, active at 10 micrograms per mil or less concentration, you could have this kind of a extensive, extended action. But if the drug would, if the drug would have a concentration of, let's say, 0.1 microgram per mil or higher, then it could have this kind of extended action. But if the concentration required would be 10 micrograms per mil, then this unbound drug would be below the threshold, and melanin binding would not help drug activity. So modeling could help in this kind of a design of drugs for melanin binding and how they would behave. We verified this unbound and free drug experiment using a high melanin binder carbonic anhydrase inhibitor. In the case of albino animals, the retention in the iris ciliary body in the albino animals was only few hours, while in the case of pigmented animals, we are talking about the range of 1 week. And also the unbound drug, free drug, could be retained at active levels for something like 6 days. So these compounds showed almost 2 weeks duration of intraocular pressure decrease after single eye drop. So this melanin binding can really contribute to the extended action. So in the final part of the talk, I will have a few comments about the interspecies scaling. So modeling can help in understanding that process. So we made experiments with fluorescent labeled macromolecules, molecular weights from 10 to 500 kilodaltons. And in rats and rabbits, as you can see, the vitreal half-life in rabbits is clearly longer than in the rats. And that is because the rabbits have bigger eyes. In the case of clearance, it turns other way around. The clearance from rabbits is faster than in rats because the clearance is related to the membranes and flow factors. Anyway, the half-life is longer in rabbits. So now if you have, for example, a rabbit study, 260 micrograms as a reference is the black line concentration predicted in the vitreous. Then if you want to have the same initial concentration in a rat, it would make this blue line so that initially the same, but it would disappear much more quickly. It would go down. And then if you would like to have the same AUC, same exposure, then you would have this red line, again quite different from the rabbit, but you would have the same AUC. If you would use the same dose, 260 micrograms, you would end up having a curve like this. And then if you would end up having at some later time point 10 micrograms per mil concentration at the same time, you would have this orange line. So you can see that it's basically impossible to make a similar profile in a rabbit and in a rat by simple injection. You can make the same exercise starting from rat reference and then making these different rabbit scenarios, and again you can see that they are very different. So in making an interspecies translation, one has to decide how to do it, but I think this kind of a simulation exercises can be quite useful also in understanding then the data. We have done some rabbit to human comparisons also, so that if intravitreal injection is given to the rabbit eye or human eye, these are rabbit clearance values calculated from literature for 12 compounds, and these 12 compounds had some data from human eyes, human vitreous injections, and they were plotted against each other. As you can see, the correlation is actually pretty good, so that the volumes of human and rabbit eyes are relatively similar. Rabbit vitreal volume about 1.5 ml and human about 4 ml. So we do not see this kind of a big discrepancy like in the case of rat and rabbit eyes. The clearance in human eye is a little bit faster because it has this higher surface area and higher flows, but the correlation anyway is pretty good, and we ended up having a scaling factor of 1.4 in multiplying rabbit dose to get a human equivalent. Later on, we did some more extensive scaling exercises, including also mouse eye and simulations of a monkey eye. So for small molecule fluorescein, vitreal half-life in different species would look like this. and for large molecule FITC dextran, it would be like these open circles. So it's possible to make some interspecies scaling, but this kind of a modeling features are really needed to do that exercise. About future challenges, one challenge is in vivo drug release in the eye, which is not so easy to do, and there's very sparse data about that. We can simulate different release scenarios, but how is the release in vivo versus in vitro is a little bit unknown. Not much data about that. For future, I think intra-tissue and cellular pharmacokinetic features would be really exciting for modeling, but that would require also some very advanced analytical systems to provide the experimental data to verify the models. And then modeling of gene, cell, and nanotherapies is still at its infancy. PK/PD models are really needed and would be very useful also in the clinical setting. And that I think requires also good data. For the modeling, there are some attempts and some papers about PK/PD models, but still I think there is a lot to be done. The PD readouts have to be also numerical and quantitatively interpreted to make the models valid. The human data is very sparse in pharmacokinetics. Pharmacodynamic data is more available, and I think the population kinetics might be the way to handle these human data, because it can handle these large number of patients, even if the data itself per patient is very sparse. Artificial intelligence obviously is emerging thing and will change the future. Of course, this is quite dependent on the data and data production, but it will be exciting to see what it will have to offer. Here are some reading materials that might be interesting. Like Paul mentioned, there is this IOVS paper on mathematical models, and then there are some other papers that I would like to mention as well. And finally, I would like to acknowledge the group members who contributed to this research that I presented, and also the funding agencies. And thank you for listening to this talk.
Well, thanks ever so much, Arto, for a really fascinating talk. I am just having a look at the questions that have been entered. So I see a question's already come in. If you would like to ask a question, please do just type it into the YouTube chat and I can relate that to Arto. So we've got a question here, which is from, apologies if I mispronounce your name, it's Guido Hanier. And the question is there an ideal nanoparticle? Sorry, let me start again. Is there an ideal nanoparticle to prolong half-life of small molecules after intravitreal injection? And are there general rules that are important here?
I think there are some features that we know. After intravitreal injection, if the nanoparticle, if it has some cationic charge, then it tends to stay in the vitreous for a longer time. So the drawback with that approach may be that if the adherence to the vitreous is too high, then it doesn't move as well to the retina. So and also it seems that there has to be some chemical features that are facilitating the retinal penetration. Some ionizable lipid nanoparticles seem to be favorable. But I think the overall to this question, there is not defined ideal nanoparticle structure that could be defined. The size is not that critical. For vitreal diffusion, you can range from 50 to 200 nanometers without major change in the diffusion coefficient. Smaller particles tend to go better into the retina, but it seems that the chemical composition of the particle has some impact also, so that there is not really very clear-cut size limit. But I think preferably I would use small particles rather than large particles. But chemical composition is important, but there is not really so defined features. Something that is very important in these experiments is that they are endotoxin free. If you look at the publications of these formulations, nobody is mentioning anything about endotoxin testing, which I find very surprising, because when we have done a couple of experiments where we had some endotoxin contamination in the preparation, the pharmacokinetics changed a lot. So then endotoxin testing is very important, and it's actually required by regulatory for market approval of such products. And then how to judge how many compartments?
Yes. So I think I can't see any other questions currently in from viewers, but if you'd like to ask another one, please do pop it in. So there's a question here. So you were talking earlier in your talk about you would try to minimize the number of compartments that you can get away with using when you've got a compartmental model. But I was wondering how you judge the number of compartments to use, how you make that judgment in terms of model complexity and explanatory power.
Yeah, we did that, for example, in the case of intravitreal injections. We had data, used also literature data. And then there is a so-called Akaike's information criterion, AIC, that can be used. And that is kind of a punishing if there's too many compartments. If you add more and more compartments, of course, the fit becomes better and better. But then this AIC is that is included, for example, in Certara's Phoenix package. It will punish you and then you can find out that, okay, one compartment model is better than two compartment model in that criterion, or two compartment better than three. In the case of intravitreal kinetics, we always were one and two compartments were always good enough. We never had to use three compartments in that case.
Sure, thank you. And related to that, I saw in one of your models that you added an extra compartment that was a delay, which I think was between maybe the vitreous and the aqueous. So
Yeah.
I guess can you explain a little bit more about that compartment? Because I guess that's not a physical compartment, presumably, but it's more of a mathematical convenience, is it? What does that represent?
Yeah, it was mathematical trick to because when the liposomes are injected into the vitreous and then they go into the aqueous humor, so there is a delay. And then in that work, we solved it by having like a virtual compartment that was kind of representing the liposomes that are on its way from vitreal injection site towards aqueous humor. In this other slide where I had this protein work, we had some equation for the diffusivity and so on. So it was handled in different way in that case. Without the transit compartment, we would end up to a situation that the liposomes would go too quickly to aqueous humor. Yeah.
And I was wondering how, like, I don't think you talked about movements of the eye, but you have, like, for example, saccadic motion of the eye as it looks backwards and forwards. How might that affect, say, the mixing of the eye or distribution of drug?
I think that has not really been studied. In principle, mixing can have an impact on. I would think that it may not, perhaps not so much on diffusion, but I think, for example, drug dissolution is affected by mixing. So if you make a dissolution experiment in vitro And if you have a very little mixing or a higher mixing, then you are affecting the dissolution, because around the particles there's a diffusion layer, a static diffusion layer. The lower is the mixing, the thicker is that layer, and then that is affecting then the dissolution, making it slower. So I think in principle it might have some influence, but I think no one has really studied. So that's interesting question, but I'm sorry I cannot give a very straight answer to this.
No, sure, that's helpful. Thank you. And I wonder if I might just close with asking. So you mentioned just at the end that the possibilities of AI. How do you see that AI could help in this area? What sorts of ways could it be applied, do you think?
Maybe integrating a lot of data, and then something that might be now with these structured models that I described might be kind of too complex somehow to handle. Maybe AI could be a way to get a lot of different data and then make something out of that. That's how I would see it. Not in a way that we use these models that I showed, that are kind of a very structure based and somehow bottom up, relying on physiology and so on. But rather, I think it might if there's a lot of data, it might be quite useful if a lot of different data will be pooled and handled with AI.
Great. Thanks very much, Arto. I'm just going to, so we'll just bring things to a close there. So I think I'll make that the last question. I'll just close by saying that our next seminar will be given by Michael Girard. That will be broadcast live at 3 p.m. British Summer Time on Wednesday, the 21st of May. Further details of all future talks can be found on our worldwide page, the link to which can be found in the About section. And do also subscribe to this YouTube channel if you'd like to keep up to date on past and future talks. So it just remains for me to say thanks again to Arto for a really fantastic talk and for his answers to those questions. Thank you for listening, and see you next time on mathematical and computational ophthalmology. Goodbye.